Good afternoon, everybody. It's a tremendous honor for me to be here today, and I would like to express my deepest gratitude to the Gairdner Foundation for giving me this opportunity to present my work. So the topic will be about CRISPR. I'm sure that many of you have heard about CRISPR. It's everywhere. It will be everywhere around us in the future. It is being used to modify crops, to modify animals, and I'm pretty convinced that it will soon be used to treat diseases in humans. But for many people, CRISPR has just 2 or 3 years. It was born in 2012 or 2013. This is not the case, and I will try to show you that it's a long story already. So this craze about CRISPR started with this paper published in Science in 2013. And this was the realization by scientists that CRISPR was a powerful tool that could be exploited to modify genomes. And in this article, Dupont and Danisco was really acknowledged for the early work that we did. So today I would like to start with some basics on CRISPR, just to be on the same page with the names and the function. And then I will describe how those features were discovered in bacteria, and I will finish with some applications, not only in bacteria, but other applications. So, basics about CRISPR. First, CRISPR is an acronym, and with this acronym, the most important word is repeats. So CRISPR defines repeats, DNA repeats found in bacterial genomes. Those repeats were new about 15 years ago because they are small, they are short, they are clustered on the genomes of bacteria, but they are spaced, they are interspaced with sequences of similar length. So CRISPR sequences, here is an example of a CRISPR. I hope you see it. No, so I will help you. The uppercase letters here constitute the repeats. So each sequence, for instance, this one or this one, those repeats are identical. Here you have, I think, 32 or 33 repeats, all identical. And it can be illustrated this way. So it's an alternation of repeats, the black diamonds, all identical, and that are spaced by unique sequences. So the small rectangles in white, where each spacer is unique and different from the others in the CRISPR locus. So this is the basic definition of CRISPR, and this is not what is used for genome editing. Associated to this CRISPR locus are some genes of more or less known function that have been named Cas for CRISPR associated. And in this example, you notably have the famous Cas9 gene. So it was really a new family of DNA that was a product of genomics, the fact of having multiple bacterial genomes accumulating. It's present in almost half of the bacterial genomes, in almost all archaeal genomes. And one interesting thing to know is that those structures were described by different teams who gave different names to those structures. And it's currently difficult when you are doing some literature searches to find those old articles, because the name CRISPR was not coined at that time. So I will just mention a few of them, but spider has been used a lot before, SR and TREP, for instance. All those DNA repeats correspond to CRISPR, but before the new name. So CRISPR and Cas function together, they form a system. And one particularity of Cas genes is the huge diversity of genes that are organized in a very diverse set across the bacterial genomes. And due to this complexity, the classification of Cas genes has been revised multiple times, and names have changed. So Cas9 before was named Cas5, CSN1, so it's sometimes also difficult to follow and to get back in the literature. Recently, it has been simplified into two main classes. So class 1 here and class 2. Class 2 define systems with a minimum number of genes, between 3 and 4, while more Cas genes are involved in class 1 systems. Those two classes are divided into types and subtypes that are defined by the presence of some signature genes. So the presence of very specific genes allow us to classify those systems into those 5 types. Actually, a 6th type has been found recently, and I'm pretty convinced that we will find more types as we sequence more genomes. The only common part of most of those types and subtypes are 2 genes named Cas1 and Cas2 in blue and green here that are found in most of the types and subtypes, but not in all. And I would like to make this comment because it's very important. CRISPR-Cas systems are not specific for species in bacteria or genera. They are really strain-specific features, meaning that within a given species of bacteria, some strains may contain CRISPR and some others not. So how does this combination of Cas proteins and repeats function together? The mechanism of action can be divided into 2 main parts. One has been named adaptation, and it's a kind of immunization that happens. So you have here a bacterial cell. I'm sorry, I switched from one side to the other. So this is a bacterial cell. This is a phage, a bacterial virus, which is infecting this cell, and upon infection, the phage DNA enters into the cell and can be recognized as foreign DNA by the cell by some Cas proteins that are able to take a small piece of the DNA of the virus in order to store it into the CRISPR locus. This happens at the leader end, so only on one end of the repeats. And by doing so, also the first repeat is duplicated in order to maintain this motif, repeat, spacer, etc. So this is the first stage, adaptation, and it's exploited through what has been called interference, whereby the bacterium uses the information stored into the CRISPR array by converting this DNA into a long RNA corresponding to this CRISPR locus, which is then processed into small interfering RNAs that more or less contain each spacer and a small part of the repeat on both sides or just on one side. And those small RNA molecules are loaded onto Cas proteins or Cas complexes in order to patrol into the cell, looking for matches with foreign DNA bearing a similar sequence to any of those small RNAs. And if this happens, so you have here the double-stranded DNA of the target, the phage DNA, for instance. If the small RNA matches one of the strands, and if in this target DNA there is what has been called a PAM motif, the nuclease, for instance Cas9, will cleave the two strands of the target DNA, leading to the inactivation of the phage DNA, and this is stopping the infection. The fact that in the target DNA there is a need for this PAM motif, just adjacent to the target sequence, is a way for the cell to avoid autoimmunity, because the same sequence corresponding to those small RNAs is present in the CRISPR array. But in this location in the genome, there is no PAM associated to the target, there is a repeat. And this is a way for the cell to to discriminate between self and non-self. I hope this is clear. So, on this information, I would like to present you some history about how this system has been found. So again, it has not started in 2012 or 2013, but it has almost 30 years of history. It started actually in 1987 when a Japanese team found, I would say randomly, some repeats at the end of one gene. And so they saw those new repeats that were unknown at that time, and they tried to study those repeats. So 2 years later, in 1989, a second paper was published by the same team, and it clearly shows that they were interested by those repeats, but they were unable to provide any explanation about the role of those repeats in E. coli. Then CRISPR has been used a lot for typing of bacteria of medical interest, and notably Mycobacterium has been genotyped using CRISPR with an approach that has been named spoligotyping. So it was based on DNA hybridization with probes corresponding to the spacers. And if I'm correct, more than 100 spacers, 100 probes, were used to classify and to differentiate strains of Mycobacterium. A few years later, the Spanish team of Francisco Morica observed that the CRISPR repeats were present in many archaeal genomes, and he proposed that those sequences could have a role in the partitioning of DNA during cell division, and that those sequences could be used to attach the DNA, the genomic DNA, to some cellular structures. I would also like to mention this obscure paper that has been lost in the battle, but here, those LTRR sequences found in cyanobacteria were also CRISPR sequences. Another example of a medical use of CRISPR, an early medical use of CRISPR, with the genotyping of Streptococcus pyogenes. So it was done by PCR and sequencing, and the diversity of spacers here was exploited to identify strains within this species. Then in 2000, the Spanish team of Morica published a small commentary about the large, widespread presence of CRISPR in genomes. So they also found some repeats in the mitochondrial genomes, but this turned out to be wrong. Actually, it was a false positive, and the biological function was still unknown at that time. Some hypotheses were provided, but many of them were wrong, including this one. So this is a paper published in 2002 by the team at NCBI, Eugene Koonin and Kyra Makarova. So they observed this presence of Cas genes in many genomes, and based on the fact that they were coding for nucleases, helicases, and other DNA or RNA interacting proteins, they suggested that it could form a novel DNA repair system in bacteria. They just missed the presence of the repeats adjacent to the Cas genes. So in 2002, the Spanish team and the Dutch team working on mycobacteria decided to give a new name to those repeats. So CRISPR was born, I think, this year. And they also described the fact that the Cas genes are very often associated to those repeats. And I think this was an important discovery in the CRISPR field. So we are now here, almost 15 years of history. And this is precisely when I started to get interested by CRISPR. This happened during a congress in the Netherlands. So I was a young scientist at Rodia Food, and I saw this poster presented by the French INRA team. They sequenced the first strain, the first genome in Streptococcus thermophilus, which was a species of interest for me because it is the yogurt bacterium. And one of my missions when I started at Rodia Food was to differentiate strains belonging to this species. And I noticed on this poster the presence of those spider and CRISPR repeats. And so that I took this picture of the poster and went back to the lab. So at that time, this species was really our white mouse in the lab. It's widely used in the industry, the dairy industry, to ferment milk into yogurt and different cheeses. It's the single grass species in the Streptococcus genus. And it's a particular species with a quite high temperature, optimal temperature for growth. And also it's able to divide very quickly. So in a short amount of time, we obtain a high level of, a high density of cells. And because they are also producing very efficiently lactic acid from the lactose in milk, they are used to get a drop in the pH of the milk in order to transform milk into something that we like, cheese or yogurt. But this species is sensitive to phages, and phages are a real problem in the dairy industry. So it's not a problem for us, DuPont, as a provider of starter cultures, but it's a problem for our customers. So I showed you this poster from INRA. The sequence that was generated by INRA at that time was not available publicly, but hopefully, luckily, we had been involved at that time in another sequencing project performed at the JGI. And the Strep thermophilus strain that was sequenced here was a Danisco strain, a Rodia Food strain. named LMD9, and end of 2002 we got access to this draft genome so that we could easily identify CRISPR sequences, design PCR primers, and start to genotype strains from our collection. And what we saw at that time was an outstanding diversity of spacers. So I will zoom in this area here just to explain. So it's a graphic representation that we developed in order to compare the spacer content across different strains. So each square corresponds to a spacer. We do not represent the repeats anymore because they are always the same. And we focus on the spacers. And based on this visual representation, we could easily group strains based on the content of the CRISPR content. So strains were grouped on the presence of common spacers. Here you have another group because they share those spacers. And you can see here an interesting separation between this group with a common part and then a different part representing two different lineages of those strains. And at the leader end, you see an even more diverse set of spacers. So we did this analysis for a lot of strains in collection. This is the locus CRISPR-1. It's the most diverse. It's also a locus that is present in all the strains, which is very convenient when you want to do genotyping. But in this species, there are other CRISPR systems. This is CRISPR-2, not present in all the strains, and CRISPR-3, absent in most of the strains. So based on this information, we obtained a very interesting region in the genome of Strep thermophilus to differentiate strain. But we quickly made a correlation with phages. So we used especially this model strain, DGCC 7710. Now it's famous because we shared it with many labs in the world. It's a nice white mouse because it contains 4 CRISPR-Cas systems shown here: CRISPR-1, CRISPR-2, CRISPR-3, CRISPR-4. And what is interesting here is that those systems correspond to the 3 major types that have been studied. So type 1, type 2, type 3. And we are very lucky because we have 2 type 2 systems in this strain. And as it was also presented by Dr. Dvorak, we had this eureka moment when we analyzed the spacer content in this strain. It was an old strain that has been commercialized in 1989. And very quickly, our customers had phage issues. And when this happens, we try to get some samples in order to isolate the phage, because this phage can be used to do challenges. It's a very common practice in the dairy industry to use a phage in order to to isolate spontaneous mutants that are able to resist to this phage. So this was done in 1990. We used this phage and selected a variant that we named 7778 that was absolutely identical at the level of the functionalities, because it's a direct beam, what we call a beam, bacteriophage insensitive mutant. And it's only 15 years later that we understood what happened to this strain. We observed that there were 2 additional repeat spacer units that we called S1, S2. And because at that time we also had access to phage genome sequences, we could make the link between those additional spacers and the phage resistance. It was a very hot period because at that time 3 papers came out suggesting that CRISPR and Cas could be acting as an immune system in bacteria, but it was only based on in silico observations. And several hypotheses were wrong at that time. So we decided to patent what we observed and tried to expand the number of cases where additional spacers could be generated. So we took the same strains, we took the same phage, and we did those challenges again. And almost every time we obtained some survivors that had additional repeat spacer units, in most cases just one spacer, explaining the gain of some resistance against the phage used in the challenge. And in some other cases, we had multiple spacers added at one time. All those acquisitions were observed on the leader end, always on one end of the locus. So we had the phage genomes at hand, and we could easily identify the region of the phage that gave these new spacers. And so that spacers could be acquired from each strand of the phage DNA. However, there was a bias for the plus strand. More spacers were acquired from the plus strand. And all the functional modules of the phage were involved in those acquisitions. And because some spacers were common to the two phages, I showed you spacer S3. S3 was acquired following infection with this phage, but it provided resistance against two phages. This one was not used in the challenge, but because the sequence was shared, the resistance was also provided, what we call cross-protection. So then we wanted to really show that it was the only event explaining this gain of resistance, so that we did some genetic engineering. We added some novel spacers in the CRISPR array, and this gave additional resistance. And when we removed the spacer content, obviously we lost the resistance. This was confirmed. It was also confirmed that when the CRISPR array was separated from the Cas genes, we lost the effect of resistance. And this last example here, we exchanged spacers between two strains, and we were able to swap the resistance profile between those two strains. So this really showed that it was the acquisition of novel spacers that provided immunity against those phages. We also did some analysis about Cas genes, and we disrupted Cas9, showing that the resistance provided by novel spacers was not active anymore, suggesting that Cas9 was important to provide this interference. And in contrast, when we disrupted the CSN2 gene here, we could still observe this resistance, but we were unable to acquire novel spacers, suggesting that this gene is involved in the acquisition step, the adaptation step. And also because in this strain there are two type 2 systems that are similar, we could observe that there were no rescue between systems. They are really specific and they function independently. So those results were published, submitted to Science. We had a quite difficult review period. But finally it was published, and it remained the most cited article in the CRISPR field until recently. And also this article triggered a lot of contacts and collaborations. So now we are here, 2007, the first biological evidence. And what is interesting is that in the following years, scientists at Danisco and later DuPont were involved in many of the important discoveries about CRISPR-Cas systems. It's all those blue dots here. So in 2008, for instance, we showed the existence of the PAM. So PAM means protospacer adjacent motif. And this has been also observed not by in silico comparisons, but by biological observations. What we did was to try to isolate phage mutants. No, not phage mutants, mutant phages. So phages that have mutated in order to escape from CRISPR. And those mutant phages were analyzed by sequencing. And what we observed was the fact that in some cases, the mutation, one single mutation, could be identified at the level of the sequence targeted by the spacer. And this disrupted the interference. But in most cases, the mutation occurred in a sequence adjacent to the spacer matching region. And this has been called the CRISPR motif and later the PAM, which is very important currently in genome editing applications. We also showed that the two type 2 systems had different PAMs, and showing that those systems are really independent. By doing some long-term coevolution studies, so challenges with phages over time, and by sequencing CRISPR-PCR products, we could show also that the PAM was important for acquisition. So it's not every sequence from a phage that can become a novel spacer. The selection is based on the presence just nearby of the PAM. It's also important for acquisition. Then in 2010, we showed that Cas9 was cleaving the DNA in a blunt fashion. It's work done in collaboration with Sylvain Moineau in Quebec. And so it was done not only with plasmid DNA, but in vivo with phage DNA, showing that the target DNA was cleaved always at the same precise location. So at that time, the Cas9 gene was named Cas5, and we showed that by disrupting the Cas9 gene, we showed that the cleavage was not occurring anymore. So that Cas9 was the only protein required to cleave the target DNA. Then with Virgis Siksnis in Lithuania, we did some experiments of transfer of a complete systems from Strep thermophilus to E. coli, so from a gram-positive to a gram-negative bacterium, showing that all the elements needed to have this interference effect were provided on this small piece of DNA. And I think this opened the door to the heterologous transfer of CRISPR-Cas systems in various organisms. Then finally, this is not our work, but this has been done by Emmanuel Charpentier. I think it's really the missing link that later allowed people to use the system as a genome editing tool. It was the discovery of another RNA, another small RNA named tracer RNA. So you have Cas genes, CRISPR. Here you have a small locus that is transcribed into an RNA, which is required to do the maturation of the small RNAs and it's needed to have the interference effect. So now all the pieces of the puzzle were together, and the next year, in 2012, Emmanuel Charpentier and Jennifer Doudna transformed a three-component system, meaning Cas9 in blue, the small RNA, CRISPR RNA, and the tracer RNA, into a two-component system with this guide RNA mimicking the CRISPR RNA and the tracer RNA. And this was a huge discovery in the utilization of CRISPR-Cas for genome editing. So this opened the door to genome editing in various organisms, and I will now present some applications of those systems. I already mentioned the usefulness of CRISPR for bacterial strain typing. I will not go further into details, but it has been used successfully not only for food bacteria but also for bacteria of medical interest. And the application which is really important for us at DuPont is what we have named CRISPRization. It's a kind of vaccination of the bacteria that we sell to our customers. It's done in a natural way because we do not want to do that through GMO approaches. So it's very simple. You start with your favorite strain. If you have a phage that is active on this strain, you can mix both and try to get survivors that you can then analyze by sequencing CRISPR loci. And when you are some luck, you obtain some variant that have additional spacers explaining the gain of resistance against this phage. And provided you have other phages that are still active on this variant, you can do the same process again using another phage in order to generate more acquisitions in CRISPR, so that after a certain times you can end up with a strain that has several novel spacers, and then you are at a dead end because you have no phage anymore. It's completely resistant to what is known. And we hope that such variants are more robust in the field, in the dairy industry. And this is actually what we observe now. So I guess that you have all eaten CRISPR-enhanced bacteria if you eat yogurt, and it's done in a natural way, so it's not GMO. We let the phages do the work, and they are very efficient at doing that. So CRISPR-Cas is really another system to fight phage infection, and we can make the analogy with restriction enzymes because it occurs once the phage DNA has been injected into the cell. At least it is valid for strains that contain, because again, some strains possess CRISPR, but some others do not. This is another very interesting application. We have called that natural genetic tagging. It's more or less the same, but without considering the resistance provided by those novel spacers. When we start from one strain, we can generate many different lineages using more or less the same phages in different orders, but at the end, the novel spacers will be different from one lineage to the other. And by doing so, we have introduced some sequences that are unique, and if we add enough novel spacers, we have a very little risk of having the same novel spacers in nature. So this is really a way to have to put a genetic label in our bacteria in a very natural way, because it's the phages that, again, do the work. And it's very important for us because we are selling cultures. It's living organisms, and anybody can take those bugs. But now we have a way to label. It's DuPont. Okay. So I cannot avoid speaking about those genome editing applications. So genome editing, I'm sure you most of all know about that. It can be done using other systems that CRISPR-Cas. It has been done, but it's more laborious. And this genome editing application is based on a break introduced in the DNA of a cell. And then it's the repair systems of the cell who will do the work. And some of those repair systems are error-prone. They introduce some mutations. And this is one way to generate mutations in a DNA. In another way, another use of this repair, a DNA template can be provided in order to direct the way the cut is being repaired. So now Cas9 is a novel tool to do those double-stranded break in the DNA, and it has been used a lot, very successively, in many different living entities. So it's a real revolution. I will not say a lot about that, but I would like to finish with this citation from Jacques Monod, Nobel Prize in 61. He wrote a book in 71, Chance and Necessity, where he says that because of the microscopic nature of the genome in human cells, it will probably be impossible to modify this DNA in order to improve human beings. And now we can certainly say that this is false. We are now able to do that. Should we not? This is another question. So with that, I would like to mention the numerous people that have worked with us. Many people at DuPont in France and in the US, notably Christophe Remo and Denis Romero. My former colleague, Rodolphe Barangou, who is also a Gairdner laureate this year. He decided to leave DuPont 3 years ago, but we are still friends. And I would also like to mention 3 collaborators, great collaborators, notably Sylvain Moineau at Université Laval in Quebec. It's a brilliant phage guy, and we are working with him now for a long time. I would like to mention Virgis Siksnis in Lithuania, and also Jean Banfield in California. Thank you for your attention.